Hi, my name is Deja Tillman. Today is April 4th, 2020, and this is lab experiment is going to be biochemical testing for microbial identification and carbohydrate identification testing. In this lab, I will incubate S. cerevisiae and S. epidermii culture, fructose, glucose, and mannitol broth to categorize each microbes into the catabolic properties. As you can see, I have labeled all 6 of my phenol vials. Number 1 is glucose. 1 is going to be with a cerevisiae, and epidermidis is the other one. Then I have number 2 vials. These 2 are number 2. I have already completed, as well as the third 2. The second one is going to be fructose, and the third one is going to be mannitol. So I'm going to show you how to do it with the glucose. So first, what I'm going to do is take my phenol red and I'll put it over the flame to make sure it's sterilized. Then I'm going to grab my glucose powder that has been provided by the lab kit. And I'll only put half of this glucose powder into the phenol red broth. So I'll put half of it in now. Okay. And then I'm going to sterilize the lid and then recap. Then I'll shake the broth until the powder is dissolved. And then I have my Dern tubes. I soaked them in alcohol here so that they're sterilized. And how I'm going to insert this into the broth is that the tube opening of the Dern tube has to be open from the bottom. So the opening of the Dern tube is going to be facing down. So then I'm going to open the red phenol red broth. Then I'm going to sterilize the lid. Then I'm going to insert the Dern tube facing down. And then reflame, sterilizing the lid, close it. And then the Dern tube, if you see it floating, you're going to have to get the air bubbles from the inside of the Dern tube and make sure it floats to the bottom. So as long as it's to the bottom, that's good. So then next, I am going to get my cerevisiae, epidermidis. Let's see, this is the cerevisiae. And then the rest of the powder from the glucose is going to be for the epidermis. So I'm sterilizing the lid of my cerevisiae culture, bacteria broth. And then I'm going to sterilize the lid of my phenol red as well. So both are sterilized. Then I'm going to get my pipette for my cerevisiae, and throughout the procedure I mark my pipette so I know identify which pipette is for each bacteria. I'm going to make sure that it's dry first. So, and then I'm going to grab 4 drops from the cerevisiae, and then I will drop 4 drops in the cerevisiae broth. 1, 2, 3, 4. And then this is going to go into the bleach because I finished the previous tubes. So that'll go into the bleach solution. And then I'm going to sterilize and recap both. So sterilized cerevisiae culture broth, and then the phenol red. So with the cultures, bacteria, I'm going to put them in the refrigerator for future experiments. And then for the phenol red broths, they all have been inoculated. So I'm going to go ahead and place them in the incubation location for 12 hours. After the 12 hours, I will observe them for carbohydrate fermentation results. And if there's no change after the incubation, I will do an additional 6 hours. After that, I will observe each broth, noting the color of the solution and the presence of vast bubbles in the derm tubes. Fermenter with acid production will be a color of orange, and fermenter with acid and gas production will be yellow, and no fermentation at all will be no color change, and it will remain red. So that concludes my experiment for today, and I'll be back with you guys with the results. Thank you. Have a wonderful day.